Hey Bio, happy President's Day. I hope you are all out enjoying the beautiful sun. It does not feel like February, it feels like April. It's nice, but it's weird. Okay, so today's screencast is Intro to Genetics. Quick reminder of the big four that we studied earlier on in the year. As you all well know, and I'm sure remember in great detail, we've got carbs, fats and lipids, and proteins. And we covered these pretty extensively in the fall term, but there's one more in the big four that we did not talk about that in great detail, and we are going to talk about in great detail now, and this is nucleic acids. And this is the molecule of heredity, and really important in the study of genetics. And so by the molecule of heredity, I mean the molecule that you inherit from your parents, which leads to traits and behaviors, and ultimately is who you are. So we're going to take a step down and look at these at the molecular level. There are actually two broad classes of nucleic acid. We have the DNA and the RNA. I'm actually going to put a picture up on the screen here. Okay, and based on the way that this is oriented, I'm going to actually switch. No. I want to move this. Okay, DNA on this side, RNA on the other. Oh, line that up there. DNA. Okay, boom. So, DNA versus RNA. As you can see, each DNA and RNA are both made up of the monomer, which is a nucleic acid. And I'm actually, before we get into this, I'm going to flash you up another picture of a nucleic acid. I'm going to do that right down here. Okay, so these are the nucleotides of DNA, as you can see here, and there are three different parts of every... Oh, this is not the full thing. Okay, wait, I'm going to get a different picture. Okay, this is a little better. So if we single out one... this is actually not great either. Okay, I can't find a good picture. I'm just going to draw it. Okay, so this is the basic structure of every, do a different color here, DNA molecule. So here we go. This right here is a phosphate group. Right here is a sugar, a 5-carbon sugar, and right here is going to be a nitrogen base. And so if you look at a nucleotide, they all share this common basic structure, but where they're going to differ is in their nitrogen base. So every nucleotide will have the sugar phosphate that makes up the backbone, and the nitrogen base will be what is variable in each of them. So going up a little further, looking back at our DNA and RNA here, you can see in this schematic, let's actually just look at the RNA because it's a little easier to see. There are 4 different types of RNA bases: cytosine, guanine, adenosine, adenine, and uracil. And that is depicted in what looks like these little sticks here. Those are the nitrogenous bases. And then what looks like a gray ribbon is the sugar phosphate backbone. So if we were to stack a bunch of these on top of one another, the sugar phosphate backbone would be the gray ribbon, and the nitrogenous base would be the sticky looking thing. Now, as you can see here, there are differences between RNA and DNA. The first being that they're actually made up of slightly different base pairs. They share 3 in common, cytosine, guanine, and adenine, but RNA has uracil instead of thymine. And that's going to be important for you to know for later on down the road. The second striking thing that's different about RNA and DNA is that RNA comes single-stranded and DNA comes double-stranded. Now, there is a relationship between DNA and RNA in terms of the central dogma, which is something that you learned earlier on in the year when we were talking about how proteins are made. It goes DNA to RNA to protein. And we're going to be looking at this more in depth. We already talked pretty extensively about this step, which is translation, RNA to protein. But we're going to be looking in greater detail at, well, actually we're going to talk about most of them more in depth, transcription. So this DNA to RNA to protein is the central dogma of biology, meaning that dogma is like a rule or a almost like a belief, but in biology we have a lot of evidence that backs up this dogma, that DNA codes for genes that are then transcribed into RNAs, which are then translated into proteins, which this is not part of the central dogma, but you can kind of add it on here, leads to traits and behaviors. Meaning all the characteristics of you, such as the fact that potentially you are tall, or you have a widow's peak, or the impulse to pee. All of these things are controlled ultimately by the code in your genes. And we're going to be talking about how, number one, how that happens, and also, number two, how we actually discovered the central dogma of biology and the history behind it. Okay, so what else am I missing here? Okay, the one other thing that I want you to stress about DNA, the structure of DNA, actually I'm not even going to tell you that now. I'm going to make you figure that out later on your own. Okay, here is what we are going to be exploring over the next couple days. There is an absurd amount of DNA in every cell. So if this is a human cell, here, that's a terrible cell, but regardless, there are 3 million base pairs in every human cell. So every one of your cells has the same 3 million base pairs. Wait, no, that's wrong. Billion. Okay, I was wrong, but I was right the second time. It's billion. 3 billion base pairs. Billion. So, that's an enormous amount of DNA. If you were to take one cell, remember how we took our cheek cells and we looked at them under the microscope, and we could see them relatively well at 40x, and then we could see them, pretty well at 100x. So that was 100 times magnification. If you took one of those cells and you stretched out all the DNA in that cell, it would be 3 meters long. 3 meters is like 9 feet, so taller than you are. That's the amount of linear DNA inside of one teeny tiny cell. So we are going to begin the next few days, number one, talking about how DNA was discovered to be the molecule of heredity, and number two, this amazing packaging problem. How do we fit so much DNA inside of one cell? There's enough DNA inside of one cell to wrap it around that cell 15,000 times. So, DNA packaging is something that we're going to be looking at. Okay, that is it. This is a little bit of a disorganized screencast, but I think you get the gist. And some of this should be a little bit of a refresher course from what we talked about in the fall. Okay, happy President's Day, enjoy the sun, and I will see you all in class bright and early, 8 o'clock tomorrow morning. Ciao.